Hello and welcome dear students. Now we are discuss here the next practical, that is simple staining of bacteria. In first practical, that is in gram staining of bacteria is essential. So in previous bacteria colorless or bacteria is colorless. Due to that, the staining or coloring of bacteria is essential to get differentiate the bacteria when we observe under the microscope. So let us see this procedure for the simple staining of bacteria. First object of that simple staining, that is to study the morphology and cell group. As we know that different groups of bacteria is available or it is observed, so we can we observe the different groups of bacteria under the microscope. We observe the size and shape of that bacteria under the microscope. So with the help of staining we can do all that morphology and grouping of bacteria. Next, which material is required for that? Bacterial culture. We have to take the infected sample which is infected by bacteria, and we have to prepare the culture of that bacteria. Next one, methylene blue, it is a simple stain. Then carbon fusion or slide, spirit lamp, wash bottle, muslin cloth, microscope and slide holder if it is essential. So that material is required for the simple staining of bacteria. Next one, the procedure for the simple staining of bacteria. So simple procedure is given here. First one is cleaning of slide. So first of all we clean the slide with the help of detergent powder to remove the greasy surface or oily surface or dirt from the slide, and after that washing or cleaning, dry the slide in air. So in first point here we have to do the drying of slides. After washing the slides we dry the slides. That means first we wash the slides under the detergent with the help of detergent powder, then dry the slide in air. After that, here sterilize the slide over the flame or both of the slide. That means with the help of a spirit lamp we sterilize the slide by passing the slide over the flame from both sides, so that whatever the microorganism which is present on upper and lower surface of slide, it get killed or sterilized. So we can sterilize the slide in second part or second step. Third one, preparation of smear. That means we have to prepare bacterial culture. A little drop of bacterial suspension is mixed in distilled water. That means first we prepare the bacterial culture. How we have to prepare the bacterial culture? We take the infected sample which is infected by bacteria, and just we have to take the small pieces of that infected part, and that are mixed with us 1 or 2 drops of water. So in that way we have to form the bacterial culture. Then that culture is mixed with distilled water, so it get diluted and placed on the slide. Then spread it uniformly and thinly with the glass slide here. So that means we have to make the smear or smear of bacterial culture here, and we have to spread that smear with the help of that glass rod. That is, bacterial culture is spread with the help of glass rod. Next one, drying. That is, allow the smear to dry in air only. Do not dry on flame. They form cluster on flame. That means we have to dry the smear by keeping the slide in air for 1 or 2 minutes. After that they form a cluster on the form otherwise here. So after drying, then fix the slide. That is fixing process. That is, warm the slide slightly by passing through the flame 2 or 3 times, so that bacteria get fixed on the slide. Here we have to pass the slide over the flame. Here pass the slide over the flame 2 to 3 times after the drying, and when we pass over the flame for 2 to 3 times, so that bacteria get fix with the glass slide. Next one, after that fixing, in that way bacteria get fixed on the slide. Then after fixing, staining. So simply we have to place 2 to 3 drops of any simple stain, for example methylene blue or carbon fusion. We have to add 1 or 2 drops of the stain on the smear or bacterial culture and allow it reacts for specific time for 1 or 2 minutes. So in that way stain we have to add on the smear. After the staining, then wash the slide gently with the help of water. So wipe out the lower surface and try the upper surface in air. Okay, so here we have to add, the staining process is completed by adding 2 or 3 drops of methylene blue or simple stain, and after that staining we have to wait for 1 or 2 minutes for the reaction to the reaction. That means the smear and stain, that is methylene blue, that are reacted with each other, and after that staining we have to wash the slide with the help of water for 2 to 3 minutes and wipe out the surface to clean the slide. So in that way, here in that way, after that washing, just we have to mount the slide under the microscope and observe under the microscope. Firstly, we have to observe under the microscope under lower magnification power. That is, we have to focus or observe the slide under 10x first of all. After that, we have to observe under the 45x, and if it is not clearly visible, then we can see the slide under high magnification power, that is 100x. So these are the different magnification powers or of object to observe that. The next, after watching or during watching the slide with the help of microscope, select the good spot of that bacteria, bring it in the center and then adjust the high power. That means first of all we have to observe the lower magnification power, and when it is observed but it is not clearly but somewhat is observed, then we have to maximize. At that point we have to increase the magnification powers from 10x to 40 or 40 to 100x depending on the situation. So when we observe under the high magnification power, so clear cut image of object is observed. So in that way we have to observe the bacteria under the microscope. So this is about the procedure of simple staining of bacteria. Here the composition of methylene blue is given, that is methylene blue 0.30 gram, ethanol 95%, we have to take 30 ml, and distilled water in 100 ml. So dissolve methylene blue in ethanol and then mix in distilled water, so to get aqueous 3% solution of methylene blue. Okay, so this is about all the procedure of simple staining. So if you have any difficulty, so you can ask me. Thank you.